Cancer Research Matters, the new podcast from Cancer Research UK aimed at our research community. My name is Phil Prime, and for each episode of this first series, we'll be speaking to some of the fantastic researchers behind the work we do. As this year, the charity is 20 years old. What better time to take us and where we go next?
Be respectful of failure. It doesn't make you a bad scientist if your idea didn't work out. Most ideas don't work out.
Sage words indeed. That was the voice of Sir Lashak Borisovich, who luckily for this podcast host at least tends to go by the name of Boris. Now, Boris is currently the chair of Cancer Research UK, as well as holding a number of senior positions at such august organisations as the University of Cambridge, MRC, and the Wellcome Trust. Medically and scientifically, Boris has had an incredibly varied career, but is perhaps best known for his early work on HPV and cervical cancer. Indeed, that is the work for which he was awarded a knighthood. Now, Boris is just simply a brilliant person to talk to get a really wide-angle view of all of the cancer research landscape. And we touch on a number of incredibly interesting points in the conversation, such as the creative act of being a scientist, through to how the various structures within which researchers operate may or may not be holding them back. But before all that, it's time to wind the clock back with Boris and find out just how early in his clinical career he was bitten by the research bug.
I started off doing basically a clinical training and was interested in research, and it was a decision that I made as an undergraduate. So my first research actually as an undergraduate was with Arthur Cochrane. He was in charge of the MRC epidemiology unit, and as an undergraduate I was associated for 3 months working with him. So my first publication was actually looking at effective exercise on peak respiratory flow rates in school children. And this was all to do with an interest then in respiratory disorders, where bronchial ability was being equated with a predisposition to asthma. And that work actually essentially disproved that as a hypothesis. We actually showed it was a normal phenomenon in many children if you study 100 odd kids. That introduced me to research, and very much I'd already decided by then that whatever I was going to do in my medical career, research would be a very important part. Only 5 people in my year in Cardiff actually did an intercalated degree, so it gives you some idea of how rare a phenomenon it was for anyone to want to do research. And I completed my clinical training and then I was fortunate to get an MRC training fellowship. I was lucky in as much as I had 2 options. One was to work on renal transplantation. I was already down a training pathway into renal medicine. But an opportunity came up to work with a chap called Patrick Sissons. And he had been a nephrologist, and he had developed an interest in viral pathogenesis. And when I started to look at the projects, I started to this issue of viral pathogenesis and protection against viruses was really important. And we wrote a proposal about natural killer cells in systemic lupus erythematosus. Actually, it would be the most boring project of all time. And I said, what's really interesting to him is cytomegalovirus, which was killing a lot of renal transplant recipients. But it would be nice to know why is it that people are particularly susceptible to cytomegalovirus and why so many were dying at that time, because we were really immunosuppressing renal transplants heavily. And he was interested, and so we really changed the whole thing, and hopefully the MRC thought that this was a reasonable change in direction, and really from then on it was history. I was really fortunate to have him as my PhD supervisor, a mentor, and he was a brilliant man and allowed me to really flourish. What he did was to let you think things through, and then he would be absolutely ruthless in criticizing what you came up with. but it was always encouraging you to try things. And that's what took me off from firstly Hammersmith and then to Cambridge. And then the opportunity came to become head of medicine in Wales. And that was quite an interesting decision point I was 38 at the time. Suddenly you were going to be put in charge of all of clinical medicine, the first time, because there was only the one medical school. And you could see that this would take you away from research. So it was a real hard search as to how much time you now have to do the clinical work and to take on board leadership as well as engaging in research.
So a bit of a whistle-stop tour there of Boris's early research career. Interesting though, isn't it, I think, that when it came to his first big clinical leadership role, research was still so important to him. And what was to happen next really was one of those periods that changes the way we think about and treat certain types of cancer.
I took the lead and set up a lab which then began to focus on papillomavirus, which was a particular interest that started in Cambridge. Something which started because of a coffee room conversation with Steve Ingalls, who was in the virology department in Cambridge. And over coffee we were saying, well, if in today's world we were to make the biggest impact possible, what would be the thing we would actually look at? And we both came to the conclusion a vaccine against cervical cancer and papillomavirus is probably something coming at it from the world of viruses where we could make a very big impact. And so with the knowledge I'd acquired about chronic viral infection, persistent viral infection in man, and how the immune system had to play a part, and particularly the T-cell immunity was going to be important in killing cancer cells, and his interest in how to create potential vaccines in a vector, which had to be a lipid vector because of the fact we were using oncogenic viral proteins to potentially immunize, led us to the sort of first trials of an immunotherapeutic in Europe in the 1990s. When you think about it back now, how naive we were in terms of what we know about the complexity of therapeutic intervention with T-cells now. The fact we even got a signal to suggest that this would actually have an impact was really a relatively miraculous event. But nevertheless, we learned a lot more. We learned a lot more about evasion, setting up international collaborations. And so that was something that I really, really enjoyed.
Now, what the wonderfully understated Boris didn't allude to there was that it was this work by him and his teams that set in motion the wheels for the development of a HPV vaccine. Then, after a huge effort with clinical trials, in 2008, the NHS adopted the vaccine and introduced a routine immunisation programme for teenage girls. And it's safe to say that the jury is now in. At the end of last year, a paper was published in The Lancet showing not only that the vaccination works, but that it reduces cervical cancer incidence rates by almost 90% in women in their 20s that were given the vaccine in their teenage years. And it's not just in the UK either. According to the WHO, some 117 countries have now introduced routine HPV immunisation programs. This has now become a global effort to attempt the eradication of HPV-induced cervical cancers. And to my mind at least, nothing demonstrates better the impact that embedding discovery science and clinical work together can have on patient impact. So, the keener eared amongst you would have picked up on something Boris said there that I think is pretty common to an awful lot of scientific discovery stories. Namely, that there's a chance meeting with a colleague over coffee, or let's be honest, sometimes something stronger, and then there's a serendipitous moment where an experiment you hadn't thought would work or you hadn't considered important suddenly throws up a result that really is important. So, given that over the last few decades Boris has worked for pretty much all the major biomedicine funders here in the UK, I wanted to ask him, how do we, as the wider cancer research community, recapitulate these often chance moments in time?
This, I think, is one of the most difficult things that research funders of any ilk. So at the moment, I work with UKRI as well as Cancer Research UK. I've been chief executive in RC and I've been on the governance of Wellcome Trust. So I really have looked at the whole gamut of funders, and it always comes back to this issue: how do you budget to enable that serendipitous moment to happen? And then how do you follow that up? How do you follow up the crazy idea that may have a chance of success of less than 5%? But it's what keeps all of us going in science, that actually you have a way out idea. How do you ensure there's at least enough resource available to be able to try something different, something that's a little bit way out? It's the exciting element. And it's the bit that makes real advances, because a lot of grants are following through, training postdocs, making sure you build up individuals, particularly in early stages, their careers, to enable them to have their own moments and to be able to go forward. So I have yet to find the perfect funding structure that enables this to happen. I think we've got some elements of it. I do think early career awards are important. I think we need career awards which are going to be over a sufficient period of time to allow people to fail and still to pick things up. When you get to 2-year, 3-year awards, it's not enough time, because you've got to expect everything to work. And we all know nothing ever works first time. It takes time. So 5-year awards I'm very supportive of, to give people that opportunity to fail, to learn from mistakes, to show what they're actually able to do, and to be respectful of failure. That, in other words, it doesn't make you a bad scientist that your idea didn't work out. Most ideas don't work out. So it is important that careers are not blighted because somebody's worked on 2 or 3 projects that didn't actually deliver the positive outcome that we would want, because negative outcomes matter, as long as we learn from them. But the system has got to be more respectful of the fact that it doesn't always work out first time, it doesn't always work out second or third time necessarily, and we still have to value the effort that people make to do this. So careers are really, really important.
Now, for many of you listening, I'm sure it's heartening to hear that from Boris. But when it comes to the career paths of especially clinician investigators, what's changed over the last 20 years?
So when I go back to it, deciding I was going to do research, and then deciding medicine was going to be the thing to follow, I went through a period where I wanted to do neurology. Then I switched to renal medicine. I'm actually qualified as a nephrologist, and then switched into infectious diseases late in my career. If any young clinician investigator wanted to do that today, the system would completely block them from doing it, or they'd be 50 before they could even look at a senior appointment. It's because we've created rigid boundaries and not allowing people to make those leaps, which I've been very fortunate to do. And I'm frustrated because it's more and more difficult to see young people coming through and having that flexibility in the structure. For all very good reasons. It's about the technical nature of medicine, the competence in being able to conduct certain areas. Those are requirements. But somehow, somewhere, we still have to build in flexibility into systems to enable people to thrive. If you look at real leaders, ignore me for the moment, you look at real leaders who have come through, they've switched. They've all followed very unconventional pathways. And yet, when we sometimes achieve those levels of leadership, what you end up doing is then creating even more rigid pathways to drive people through. And that's something that I do think we have a big responsibility for looking and making sure that opportunities remain open for people to switch, to change, to allow new ideas to be able to flourish.
Now, there's pretty much a general consensus now that for cancer research to be at all effective, it needs to be multidisciplinary. And many modern research labs are indeed just that. And while that's clearly a positive thing, perhaps this multidisciplinarity needs to be rooted more at the level of the individual.
I think that's really is very important. So you're placed in a multidisciplinary environment, but it still very often means that people don't necessarily switch direction themselves. They work in a team which has got different disciplines in that team. What I'd like to see is within that team to make somebody who is a scientist who may have a particular perspective actually take more broader responsibility. You see this in engineers who suddenly become bioengineers because they've seen a greater biology perspective. So yes, I'm optimistic that some of these changes will happen in science. The problem comes for clinician scientists where you're stuck to a very rigid structure because of the needs for patient safety and competence in your clinical practice, and particularly in so-called craft specialty surgery and elsewhere, mitigates against you having the freedom and the time to be able to undertake some of those exciting research dimensions, and therefore there is a risk we could be losing some of these entrepreneurial individuals who actually have got so much to add to our field. Now, there are many great people thinking about how we get around this. There are reasons to be optimistic that people are realizing that this multidisciplinarity, this capacity to work in teams, this capacity to collaborate effectively, and that it's okay to switch. Don't pigeonhole yourself. You can actually, as an investigator, take on all sorts of aspects. I think that's great, and I think it will help us. We also have to learn from other specialties, because team science is something that is still in the life sciences somewhat blocked by this obsession of who's first author. I mean, the number of papers with 7 first authors now, who's first author, who's last author. You know, when we see the paper on the Higgs boson in the field of physics, there was something like 300 names attached to it. I didn't see a single career being blighted amongst all the species by whether they were 107th or 108th, because everyone played their part in a team. And I think in life sciences, we've got to wake up to this, that the teams are getting bigger, and therefore collaboration, participation, and clarity how people participate.
Much appreciated for
Their role.
that, Matt. Much appreciated to you.
We consider academic promotion or
Because I do actually believe it is a good cause to do. So much appreciated for saying.
Was he first author? Was he last author? I mean, frankly, in the greatest scheme of things, if it was a good discovery, who cares?
Because when it comes to cancer research, it will forever be the main charity that I support,
So I remember getting into
no matter what. So again, much appreciated to you.
CDC and ICRF, they decided they were pulling out the viruses. That was a decision. Immunology was not seen to be very important, all based on a lot of failed work. So now when you look back as to what we're seeing with HPV now being seen by the WHO as being
Yeah, I can't like.
eradicating a cancer to deliver womankind a real scourge of a disease, but for the WHO to mention that now as an ambition
Just keep on.
to eradicate that actually states just how far we've
Much appreciated for saying that. It's really
being a
good.
Cinderella discipline just kept lying.
True, it is like.
Now to checkpoint inhibitors, immune checkpoint inhibitors.
I just want to
acting on disease.
find the bearings at the moment.
Discussion on cancer vaccines and inducing immunity to individuals' cancer, the debate about whether you try immunotherapy.
Yeah, I get that, like.
Salvage therapy, the cost of it, why it fails in others. Open up a whole field which is.
And much appreciated for the likes there as well, Matt.
Where it was in the 1990s, by decisions that were taken and were quite rational to have been taken at that time. But the ability to adjust to those decisions and to move with the changing.
Because any, much appreciated for the likes as well. Because anything I get, and I do donate to cancer research. It's the main reason why I've got that little donate button on the screen. Because I figured why might as well cut out the middleman. and let other people donate to the charity themselves.
Why does cancer metastasize? Let's be honest, we are pretty good at controlling local disease, and in many cancers, I mean, it's the metastases that will actually kill.
Don't that,
And yet our control of
I'm going.
metastatic disease is not great. So when do they arise? We've got some information that the mutation rate within metastasis may be less than those from the primary lesion that we actually see in cancer. So,
Well, I do appreciate all these likes you're sending them out. Much appreciated.
Cancer cells is another one that these are ones that really bug me. How does a cancer cell lie dormant for all those years? In viruses, this was a big question about viral
It is quite a massive pattern that.
What causes it to wake up again?
No,
And then other
one of the probably the biggest out there, like.
Still we come after our success on the smoking program really works. It's going to become the commonest cause. What are the mechanisms? Why only 13 cancers are associated with the obesogenic environment? This is hugely important to understand.
Because I believe
Completely novel therapeutics
and help now where you can, where I can.
Environment of individuals and how cancers can interact with that. So, yeah, these are my sort of cancer obsessions. The fact that we can begin to predict driver mutations, but a series of studies that have been conducted.
And if I can help out cancer research, however I can, I guarantee you, I will.
A is followed by B, or B comes first followed by A, can give you a completely different phenotype. Why? So understanding the mechanisms in cancer really, to me, are absolutely fundamental. And if we want to make real advance understanding these, and then harnessing the therapies around our greater understanding of cancer, does actually pay dividends. The last is something that goes back to my time with Archie Cochrane. And that is the whole system approach. If we come up with new discoveries, do we actually have a system that can actually adapt to make that available to the majority? Archie Cochrane wrote a book called Effectiveness and Efficiency. But when I was with him,
So what is everybody's plans for the day and what are they doing up
he asked me
to?
at one point, he said, what do you think? He said, what puzzles me is if we can't deliver something efficiently, what's the point of proving its effectiveness? And that's where it's always had me thinking that there is a whole system that has to work in terms of translation.
This size, usually,
Greatest number.
is a bit of a pain in the ass.
So there we have it. Thank you so much for listening this far. Thanks to Boris for being such a brilliant guest. Please do keep your ears peeled for the next episode where I chat to our outgoing Chief Scientist, Karen Bousden. But for now, it's time to leave the last word with Boris, with a powerful reminder that there is so much more to you as a cancer researcher.
Much appreciated for all the likes.
Just churning out experiment after experiment.
We share more with the creative arts in some ways than we do elsewhere. Science is a creative activity. It is not merely following an algorithm that's going to get you to X and Y, which is one of my anxieties in the way that science is funded today, is a belief from the experience of COVID that merely if we put a big enough team together, throw enough money at it, we will end up with a solution. There are no guarantees. The vaccine works that even underpin the AstraZeneca vaccine goes back to the 1990s by identifying this as potential vectors.
Oh no, the silver's gonna need a second coat though.
Use that backbone.
Usually the bone silver that usually takes most coats to do.
Always building on this mountain of underpinning research that enables you to make the next leap forward. Most of the research that is undertaken is going to find some utility somewhere. I'm a great believer in that statement that is attributed to Porter, but is well known that there's only one type of science, and that is applied science or not yet applied science. And if we do good good science, the applications will come forward, as will the benefits to human health that stem from it.
That is
Hello and welcome
a gentleman. I don't know if he's still in or not,
podcast from Cancer Research UK.
but that gentleman is
My name is Phil Pine, and
one of the
in this
most
episode,
nicest
We'll be
lads.
speaking to some of the
Yes,
fantastic
there is.
researchers behind the work we do. Because this year the charity is 20 years old, what better time to take
Oh,
stock
doo
and see
doo.
where we've been and where we go next.
And we got very, very close. You know, there was a time where we thought we got it with good sortedness, we solved cancer. At least for 50% of people.
That was the voice of Professor Karen Bowsden. Karen is a group leader at Francis Crick Institute and will of course be known to many of you as up until recently she was CRUK's chief scientist. Now as she has come to the end of her tenureship, very grateful we all are too for her sage leadership, this is the ideal time to get some of her thoughts on her field of tumour suppressors. Famously of course she's done a huge amount of work on P53 and to look at some of her other work on cancer metabolism. But we also touch on many other things, such as her role in the development of the HPV vaccine and how the life of a researcher has changed. So sit back, all you do, and enjoy this conversation with Karen, which started officially
It started very early on as a microbiome
Okay, doesn't mind the code change. Have a different feeling. This is going to take.
So that's how I got cancer research, and then I moved around to various different areas. So when I was
A few comments.
on identifying oncogenes, RAS oncogene, and then I moved to America to do a postdoc where I worked on papillomaviruses, and then I came back to the UK and I worked on P53 for the longest time. So I guess P53 is really where most of my work has been. And then because of P53, there was a link with metabolism. I started to work on metabolism. So metabolism has been the most recent iteration of my interests. P53 has been quite interesting. So I think there was about 10 years maybe where P53 has been described as a protein and no one was interested in it. So no publications that really happened. And then a couple of things happened. Carol Pries and Arnie Levine and others showed that P53 was a transcription factor. And a number of people, including them and Moshe Oren, showed that actually the P53 that everybody had been looking at was a mutant variant, and wild-type P53 stopped cells from growing. And Bert Vogelstein showed that P53 was mutated in enormous numbers of human cancers. 50% of tumors have mutations in P53. So taking all that together, people kind of thought, okay, here's a protein that stops cells from growing, stops cancer cells growing, and it's lost in almost every cancer. This has got to be interesting. And then there's sort of a massive explosion in the number of people who are interested in P53. David Lane did some really lovely work. And after a while, during that period, we suddenly thought we knew what it was doing, right? We understood P53 responds to damaging signals, DNA damage, and it gets activated and it kills the cells that have sustained this genotoxic damage. So basically, cells that have alterations that might make them into cancer cells get killed. And so you're kind of eliminating everything that's a potential dangerous cell, and that was it. And we all understood P53 and it was wonderful. And then, because there were so many people working on it, they carried on working on it, and everything got much more confusing and complicated. And so now, there are lots and lots of things about p53 that we clearly don't understand. We know that it's a transcription factor, but we know it has transcription-independent activities. We know that it can kill cells, but it can also have many, many other effects on cells, including helping cells to survive, which is paradoxical. We know that p53 is mutated in many cancers, but under some circumstances, we can see that those mutations don't just inactivate p53, but they drive new pro-oncogenic effects. So we flip p53 from a tumor suppressor to an oncogene. And all of that has become really complicated to try and,
Yeah, this is going to need a couple of quotes.
So, teasing apart p53's role in cancer and how we might utilise that potentially to develop some kind of therapy is then a very tricky business indeed. And it really does seem to be a case of the more we know, the more we realise we don't know. And that, of course, is not an unusual story in biological research. But there are reasons, as we'll discover later, Karen, to be cheerful on that front. Now, on the subject of complexity, that is something that I, for one, always associate with metabolomics. And while that may be true, says Karen, it's always worth remembering that
this is kind of the renaissance of metabolism. Back in the 40s and 50s, when people were first working out metabolic pathways and Hans Krebs and all the other guys, they did an unbelievably remarkable job of figuring out the fundamentals. And what really impresses me is you go back and everything that they said is right. They got it all right without, single-cell sequencing or transcriptomics or proteomics or all of this stuff. On a shoestring, they got it all right. So we have a very strong basis on which to build our understanding of metabolism in cancers. And that's quite rewarding, to go back to these old papers and think these are the things that they describe. Of course, it is more complicated, and we do know more, and it's much broader. But the fundamentals are all there. I guess the way the fundamentals of genetics were established in those early days. But yeah, you're right. I don't think anything gets any easier. Having said that, though, one of our ultimate goals is to help patients. And I think that the level of understanding that we have with many of these pathways is sufficient that we can start to develop and devise new therapeutic approaches. We still need to understand more, and I think that becomes very clear when we're thinking about, how do we get resistance, or how do we properly, combine therapies, and what is the expected outcome of treating a cancer patient with therapy X, and how will therapy Y work even better. So there's lots still to know. But I think that, the advent of immuno-oncology is a fantastic example of that. How understanding how immune responses to cancers are dampened has given us this fantastic opening into how we can target that. And it's incredibly successful. It's just so exciting.
Now, this brings us nicely up to the distinctions between goal-directed and curiosity-driven research. Now, it's something I really wanted to talk to Karen about. She's had a very successful academic career. She's had a number of scientific leadership roles. So I really wanted to know, for a field like cancer research, where does that balance lie?
Well, I think you can't do one without the other, really. I think that we're required to understand the fundamental mechanisms of what we're studying. And without understanding that, which you could put into the blue skies curiosity-driven research, but it isn't really. It's simply wanting to understand at a very fundamental nuts and bolts level how something works. Without knowing that, you're never really going to be able to devise a proper intervention. You're not going to know what the response to your therapy is going to be. You're not going to know why some people respond and other people don't. You're not going to know why people appear to respond and then relapse. So I don't think, I don't have any issue in, I don't see these as two different things. I see them simply as necessary components of one push forwards.
Yeah, interesting, isn't it? I wonder if that's a mindset that develops over your career as a researcher. perhaps as an earlier career researcher, you really are just following things that interest you.
I think that's true. I mean, obviously, the older you get, the more interested you become in practical applications. Not necessarily because it affects you directly, but because you think it would be just really nice if something I did made a difference. You know, it's a sort of legacy thing. You know, it's nice to be in the textbooks, but the story I always tell is my contribution to papillomaviruses. So I worked on papillomaviruses because I'd worked on rats. And so because I'd worked on an oncogene, and papillomaviruses are being, you know, considered as a driver of cervical cancer, I thought, well, you know, if they're causing cancer, they must have oncogenes. So I'm going to use what I did to describe, you know, to find RAS oncogenes and find the oncogene in papillomavirus, which I did successfully. And I was able to identify 2 of the genes in papillomavirus as having the potential to make a normal cell into a tumor cell in an experimental system. So one of these proteins binds p53, and that's how I got into p53. But, you know, I didn't work on papillomaviruses for very many years, but I did make this teeny tiny contribution to supporting the notion that papillomaviruses cause cancer. And then, you know, wind forward 20 years, and my daughter and her friends are being vaccinated, you know, with something that's going to stop, clearly, you know, the evidence is there now, stop women developing cervical cancer. So, you know, it's just absolutely thrilling. I think it couldn't be better. So things like that, you really want to feel that, you know, everybody wants to feel that what they're doing is worthwhile, and of course the pursuit of knowledge is always worthwhile. But then the added icing is that, you know, it can be a practical value as well.
Now, obviously, I couldn't resist. Does she tell her daughters about her involvement, or does she remain silently heroic?
Yeah. Oh yes, I tell them.
Now, time to get back to Karen's work on P53 for a moment. As she previously mentioned, the field had attracted an awful lot of attention because of the potential of P53. However, the data-rich environment also adds to the complexity. So, how does Karen think about the last 20 years in the field, and where does she see research on P53 going?
I think P53 has been very, very frustrating in that we do have quite a good idea of what's going on when you activate P53 and why that doesn't happen in tumours. And, you know, we have one animal models where we can show that if you could reactivate P53, you would have a very strong anti-tumour effect. And yet, there isn't really any good therapy that's directed to P53 status in the cell. And, you know, it's been a long time since we had access to small molecules that actually stabilize P53. And, you know, the hope that these would just be incredibly good therapeutics for those cancers that retain P53 has kind of never really lived up to its promise. And there are various possible reasons why that is, which, you know, you hear and then people say that's not true. There's still a lot of, I think, confusion around. The fact is there are a few, you know, inklings of efficacy, but in general it's not been the massive success that we all felt it would be, which has been really, really, really frustrating. And, you know, there's still a lot of very good people working on this and trying very hard. And I don't doubt that at some point it will work. But it's just been more difficult than it should have been. The theory is perfectly sound, but just actually getting a practical solution has been more difficult than we imagined. And we got very, very close. You know, there was a time where we thought, yeah, we've got it, we've sorted this, we've solved cancer. At least for 50% of people.
So, the ongoing enigma of P53 aside, academically speaking, what else has changed for Karen's lab?
I'd say the big shift in my lab over the last 5-6 years has been to take our focus away from actually the cancer cell and think more about the environment. And that's, I think, been a big shift in cancer research globally. You know, this understanding that cancer's not enough, they don't live in isolation. They're a community of different types of cells. And, you know, our particular interest is back against the metabolism and nutrition side, which is how does that community of cells feed itself, feed each other, and what's that interaction do? How do cancer cells manage the whole journey of moving from a primary site to a metastatic site, all the stress that's involved to the cell in trying to survive that? I mean, we know very few cells do survive it, but enough do to cause problems. How do they measure? How does a cell that came from the pancreas manage to lodge in a lung and then proliferate there? You know, the nutritional environment is probably quite different. So what is it that allows that cell to do that? Why do some tumours metastasize to some organs and some tumours metastasize? So we're much more interested in that kind of environmental question, seen through the lens of metabolism and nutrition.
So, time to shift gears just a little and talk about life as a researcher. What that's been like for Karen, how she's seen that change over the last 20 years.
For me, I'd say that life as a scientist is a privilege, really. You know, I get to do what I enjoy. I get to interact with the smartest people. I get to work on problems that are really interesting and intriguing and yet beneficial and useful. So I can, you know, I love it. I can't imagine ever doing anything else. I mean, things have changed. You know, the way the technologies available to us, of course, have changed enormously. You know, when I started my career in cancer, that was pre-sequencing. So I remember I started sequencing when I was a postdoc on, you know, big sequencing gels where you could read maybe, you know, 200 or 300 base pairs. But that's, I think the environment has really changed, and just the ability of us to do things, you know, the microscopy, the imaging, the sequencing, the proteomics, just our ability to really delve into, you know, biology and measure it is just astonishing compared to where we were when I started. So that's a big change. But other things don't change, you know, the way we work, the ambition, the tears, the happiness, you know, the ups and downs. It's all still the same. You know, we send our papers off, they get rejected, we cry, we dust ourselves down, we send them back out again. I mean, I think research is such an interesting arena in that most of the people that I know, what they really, really want is for their colleagues to think well of them. So it's a real strong driver to you to have something interesting to go to a meeting and be able to say to your colleagues, I found this interesting thing, for them to go, oh, that's really interesting. I mean, this is just you know, a really important thing to many scientists. And it's, you know, it's a reward in itself. So I you know, I don't know, I find people in my lab, and, you know, that I come into contact with more anxious about career development, career structure than I ever was. And maybe it was just stupid. It never occurred to me to worry. I remember when I got my PhD thinking, you know, I've got a PhD, I'll probably always have a job. And I feel that people get more anxious about their future now. Maybe it's more difficult, maybe the competition is great, so maybe it was easier than my day.
Interesting. So, that said, has Karen any tips for early career researchers?
Yeah, people ask me this a lot. And, to some degree, I mean, you have to accept that your own success is largely driven by luck. You're in the right place at the right time, the work went well. But I really, really encourage people not to get too stressed about these issues of where am I going to be in 40 years' time. I really encourage people to take the risk in their careers and to follow the path that most excites them. Because I think the biggest gift, one of the biggest gifts we have in life, in the way that we have to live, is to have a job that we enjoy. There's nothing more depressing or demoralizing about not wanting to go to work. And I think we've all done it, I mean, I used to work on the cheese counter at a wine fair, and I could not wait to get home. So I think that given so much of our time is spent at work, and given that science has so much to offer, I would want people to choose an option for the next step in their career that excites them. I want them to choose with their hearts and not with their heads.
So, just before I leave the final words to Karen, it's just time for me to say thank you to her for talking to me for this podcast. And on behalf of all of us at COUK, thank you so much for your time as chief scientist with us. As for the rest of you, thank you so much for listening as always. Do have a rummage through the show notes because I will put some links to some interesting articles about Karen's work in there. But for now, what would you like to know that we don't know now if we were to have this conversation again in another 20 years?
I mean, I think, what we don't, we've worked a lot and we know quite a lot about the middle part of tumour development, which is why and how does the primary tumour develop. What we don't seem to know very much about are the two ends. We don't know very much about what is it that makes a normal cell into a tumour cell. And again, there's an example of where we thought we understood it, and now we know we don't understand it. Because it's not just the genes that are mutated, because we know we've all got loads of cells that have mutated tumour suppressor and oncogenes, and they don't develop into cancer. So that's probably back down to the environment again. So that would, I'd like to know more about that. I'd like to know, because I think then we could make much bigger strides on cancer prevention, which of course is much more sensible than trying to cure people who have cancer. But then we don't really know very much about the other end either, which is, how do cells survive metastasis? How do they know where they're going? How does a primary tumour affect your whole body? So that basically, some of these metastatic sites are already primed to receive the tumour cell. I mean, it's scary. It's science fiction. But I think we need to understand all of these things if we're going to help patients that present with cancer. Because we know that, particularly for some tumour types, the real problem isn't the primary tumour. The real problem is that by the time people realise they're not well, the cancer's already spread. So lots of questions about metastasis and metastatic spread that we don't understand. I would like to know much more about.
Hello and welcome to Cancer Research Matters, the new podcast from Cancer Research UK aimed at our research community. My name is Phil Prime, and for each episode of this first series, we'll be speaking to some of the fantastic researchers behind the work we do. And because this year the charity is 20 years old, what better time to take stock and consider where we've been and where we go next.
The lessons that we're learning from children's cancer really spawning what could be very transformative understandings of adult cancers. And that's actually it. The heart of that is biology.
That was Professor Richard Gilbertson. Now, Richard is director of the COUK Cambridge Centre, as well as the COUK Children's Brain Cancer Centre of Excellence. Academically speaking, he's best described as a paediatric oncology clinician scientist, and his work has been dedicated to furthering our understanding of childhood brain cancers. Now, this podcast also happens to be going live in September, which is Childhood Cancer Awareness Month. So, a perfect time to talk to Richard about where we've been with research on childhood cancers, where we might go, alongside some of the incredible breakthroughs his lab have made on the origins of childhood brain cancers. Now, always looking to get the most out of our guests here on the Cancer Research Matters podcast, we also get into some fascinating, wider angle topics, such as self-testing, diagnosis, and where healthcare in general might be headed. Now, Richard's research story began way back when he was a clinician, and it was in a particularly profound way.
So it was back in my first week of medical school, and actually I was just assigned to a ward. I was in Newcastle, and my first ward was a paediatric ward, and there was a little girl there dying of a brain tumour and medulloblastoma. And I got to know the family in the 2 weeks that I was there, and I watched her die, unfortunately, and it had a profound effect on me. I still don't understand particularly why, but other diseases or patients. But at that point in medical school in the mid-80s, I sort of vowed that I would work on that disease, and I have done for the last 30 odd years. The thing that really affected me, that made me stay in the lab as opposed to the clinic, was something that a good friend of mine, Nigel Stout, who was my best man on Mary's, actually challenged me when again I was a medical student. It was in our first year. And we were in the pub, and Nigel sort of was a bit drunk, but then he sort of stood up and he pointed at me and he said, "It doesn't matter what you do with your life, you've just got to be responsible for 10% reduction in mortality of a disease." And then he collapsed. And that sort of stuck with me at the time when I've been thinking about medulloblastoma. And that short statement, which was a sort of very silly thing at that point in time, I always kept thinking about it when I was thinking about what I would do for my career. I realised that actually the place I would get the 15% that he challenged me to would actually be in research. It wouldn't be particularly just in the clinics, as important as that is. And so that's why I sort of dedicated my life to research. It did have a profound effect. I mean, I don't see patients anymore, but they continue to impact my research, and everything we do in the centre or in the department where my lab is very much focused on making the lives better and longer for patients.
So, all our research into childhood cancers as a field is both right. Obviously, in many regards, it's very well established, but it is still relatively small. There are reasons, says Richard, why this is likely to change. So, over the course of his research career, how has he come to think of paediatric oncology as a field?
In many ways, it's more advanced than adult cancers. I mean, the reason why it's perhaps received less attention is it's rarer and much rarer than adult cancers, obviously. Although cancer kills more children than any other disease in the developed world, so it is the biggest killer of children by disease. And also, because it's a smaller field and fewer people go into it, the funding is historically less. The other thing which has happened with children's cancer is that, like any sort of field in science, not just in medicine, but it's driven and propelled forward by major discoveries, so there'll be a sort of milestone or a sensible discovery, and then people will sort of get organized around that, and it will accelerate the field, but it's discovery within the LBS century.
Here is the family Jane. Distracted him. Oh, there. Right, is that on Atomic Drive? Start. Oh, I need a.
It's sort of punching way above its weight, largely because of that collaborative effort that's sort of brought about by that person. So the so well known that civil field has always been a very established community, and in many ways, dare I say, a more mature community in the way it acts and collaborates. And however, what I would say is that the lessons that we're learning from children's cancer and the education of children's cancer really supporting what could be very transformative understandings of adult cancer. And that's actually the heart of that is biology. So whereas we feel as breast or prostate or lung cancer, you're often studying the difficult situation.